seminar in the series entitled The Many Facets and Faces in Health Informatics. In addition to the audience here, I'd like to welcome our web viewers and Ontario Telehealth Network sites. Just so that you know, this seminar is being recorded and is being broadcast over the web. We ask that you hold your questions to after the seminar presentation so that we can get a real or virtual mic to you. A very warm welcome to our guest speaker, Dr. David Hill. David is a professor in the Department of Physiology and Pharmacology at the University of Western Ontario, and he is also the scientific director of the Lawson Health Research Institute. Dominic will introduce David shortly. I'd like to acknowledge our seminar series sponsors, Borden Ladner Gervais, McKesson, and Healthcare Information Management and Communications Canada. We could not host these seminars without their support, and we're very grateful to them. Before we begin, I'd like to announce our next seminar in this series. It will be held on Wednesday, February 27th, and our guest speaker, we must have a focus on University of Western Ontario. Our guest speaker is Dr. Richard Kim. He is also a scientist at the Lawson Research Institute. He's the chair of the Division of Clinical Pharmacology and the director of the Center for Clinical Investigation and Therapeutics. The title of Dr. Kim's seminar is Personalized Medicine, Today and Tomorrow. This new seminar series will look at the many facets of health informatics and some of the key people involved in these areas. Over the next several months, our guest speakers will talk about personalized medicine, as I just mentioned, medical imaging, information and communications technology to support our health system, and the role of geographic information systems in healthcare. So I'm really looking forward to this series, and I hope you will all come back for each of these seminars. So our agenda for today is as follows. Professor Dominic Covey, founding director of the Waterloo Institute for Health Informatics Research, will introduce our speaker. David will speak about 45 minutes or so, somewhere in there. We will then have a facilitated discussion. We'll ask, as I said, that you hold your questions to after David's presentation. Please signal if you got a question, and we will get the mic to you. And just to make sure that we record your question, please make sure that you hold the mic up to your lips, please. So now may I introduce Dominic Covey, who will introduce our speaker.
Well, good afternoon, and also Happy New Year. It's good to see you here, and the ones I cannot see on the web, welcome. We're looking forward to this year. It's a different orientation. Dr. Hill is not a health informatician himself, but he's running an institute of research. We thought it would be very, very interesting to hear about the nature of the research that's going on and the types of informatics forward-looking that's being done there. We already know he's scientific director of the Lawson and integrated vice president for the London Health Sciences Centre in St. Joseph's Healthcare in London, professor in the Department of Medicine. He's actively involved in national and provincial health research advocacy as a board member, for example, of the Research Canada. He has over 200 publications, and his research centers on the generation of new insulin-producing beta cells in pancreas as a strategy for the reversal of diabetes. I've known Dr. Hill a lot longer than we've known each other. I heard much about him, all good. I was very pleased several years ago to have a chance to speak with him and to learn a bit more from some of the people. Famita Gwadri Sridhar is here as well, joining him, and I'm sure ask questions. She would be happy to share things as well from Western. The work that's going on at Western is world-class health research, and the building of the foundation of informatics under it, I think, will be an interesting piece of work that goes on, and it should be observed by all of us. So I'm looking forward as things kind of roll out in that area. Welcome, David.
Thanks very much, Dominic. It's a pleasure to be here at Waterloo. Many of you may not realize, but certainly over the last 1 year, 2 years, we've been making increasing linkages between the Academic Health Sciences Centre in London and Waterloo, and looking for interfaces where we can productively synergize and work together. And certainly health informatics is turning out to be one of those extremely promising areas. It sounds like this is a good choice for a first lecture, because in fact I'm going to touch on the range of types of information that we can gather through a health research institute, and why there is an increasing demand for sophisticated health informatics tools to mine and make the most of that information. And in fact, I will show just a couple of slides from Richard Kim's program, and he will then, I'm sure, give you a much fuller account in the next seminar around the area of personalized medicine. So I've entitled the talk Health Informatics, Critical Support for Clinical Research, which really says that our approach to health informatics is as a tool. Okay? We know that there have to be experts. We applaud the excellence that is in centers like this, and the discipline of computer science and the purity of health informatics principles and development and applications. From our point of view, we're very much the user. We have incredibly sophisticated piles of data that are accumulating in a way that we would never have dreamed possible a few years ago. And our challenge is, how do we make the most of that data? How do we sift it? How do we sort the relationships? And how do we pull out of that value that we can apply to healthcare provision, particularly in a hospital setting, because we're a hospital-based research institute. So first of all, I just thought I'd give you a few basic stats about our institute. So the Lawson Health Research Institute is sponsored by 2 hospitals in London, London Health Sciences Centre and St Joseph's Healthcare. So we represent all hospital-based research in the City of London. We're fairly big by Canadian standards. Our throughput of grants and contracts, the vast majority of which are external funding, is around $60 million a year at the moment. And that income and that work is generated by a core group of around 140 scientists. And I define that by an individual who spends the majority of their working day thinking and doing research. Now, of course, we are supported by a cast literally of hundreds of associate scientists, because all of our physicians working in the hospital sector have faculty appointments at Western and also have a mandate to do research. Often that is part-time. It may be clinical trials portfolios, it may be health outcomes research, but nevertheless they are part of our community. We at the moment have a hiring rate of young scientists of about 40 over the last 5 years. So at any one time we're aiming for about a third of our scientists to be in the first 5 years or so of their career. And I think that's really essential, to have this succession plan and this new blood coming through constantly. Because at the end of the day, certainly, people can get stale if they're not challenged with exciting new ideas, and it's very important for our institute to always be in touch with fresh ideas. We're a major training site. Most of our graduate students are associated with Western, but we have 190 graduate students training. Everything from basic science through to population health research. In CIHR terms, we cover everything from Pillar 1 to Pillar 4. We also have about just over 100 postdoctoral fellows and clinical fellows from all around the world. We don't just depend on grant income, as no university or research institute can these days. We have a very active business development office, which currently is generating income of around $2 million a year, both from spin-off companies, but also from royalties from intellectual property. In terms of productivity, we are monitoring productivity and quality of our research fairly carefully. Obviously, you can't do it until a couple of years have lapsed and people have actually read and cited your work. But for 2005, we were just shy of 14,000 citations for, at that time, a core of 112 scientists. And certainly, if you look at our most intensive group of scientific leaders, then they're sort of hitting the standard, which it is around 12 citations per paper per year. Also, we are a major site for clinical trials activity. There's 1,800 active clinical trials going on through Lawson, which is a huge volume of development work, and obviously, because of that, a huge partnership with the pharmaceutical sector. So just one example of one of our spin-off companies. This is a company called Fralex. Its technology was to apply magnetic fields to actually alter biological systems. So we're used to thinking about the potential harmful effects of magnetic fields living under power lines, etc. In fact, some configurations of magnetic fields can be beneficial. We have a large IP portfolio that shows that magnetic fields can fire receptor systems, can give biological endpoints, and you can harness and use these. And a main indication of this company was for analgesia. So by wearing a headphone with pulsed magnetic fields, we can actually induce an analgesic effect, which is very effective in chronic pain. This company was sold to the private sector last year, floated on the TSX, and is now doing very, very well. Our focus areas are all of the areas you would expect academic hospitals to be active in: vascular health, chronic disease, surgical advances, critical illness, so emergency medicine, children's health research, cancer, imaging, aging, rehab and geriatrics, and mental health. We also have a very active outreach. One of our programs is a Center for Probiotic Research, the use of beneficial bacteria to colonize body environments such as the gut and keep harmful bacteria out. And one of the most useful environments you can translate this research is in fact in the developing world, which is stricken with things like childhood diarrhea, AIDS, etc. And so we have a clinical trials network in East Africa, which is based in Tanzania, and is actually looking at novel probiotic projects to maintain the health of communities. And through this project, we've been able to partner not only with governments in East Africa, but also with major companies like Danone, the French food manufacturer, who are partnering not only to bring science, to evaluate science in Africa, but actually build businesses, nutraceutical businesses that helps the economy of the local population. So why, with this sort of background, do we need to worry so much about health informatics and identify it as a priority need? Well, basically because of the depth, the complexity, and the potential of the data that we can now collect, and really has just exploded on us in the last 5 years. We would never have anticipated the volume and the detail of the data that has emerged through various technologies. And what I'd like to do today is firstly show you some examples of that type of data, and then show you how we are going to approach organizing health informatics to take advantage of that, not just at Lawson or at London, but with a regional Southwest Ontario approach. So starting with imaging. So imaging traditionally has been imaging anatomy. So, X-ray, high-definition X-ray, CT, MRI, it's all about looking at anatomy. But now imaging has gone beyond that. Instead of imaging just physical anatomical structures, we can now image molecules, cells, even gene expression non-invasively. So instead of simply being a domain of physics, it's now become a partnership between physics, molecular and cell biology, and chemistry, combinatorial chemistry, to come up with novel molecules that would act as targeting agents for imaging signals. What this enables you to do, as I said, is to actually non-invasively image molecules and genes deep in the body non-invasively. And this really now gives you some additional insight into the nature of disease. So now you no longer have the potential to look anatomically at a tumor, but to actually classify exactly what type of tumor that is, and what are the aberrant genes that are being expressed in that tumor non-invasively. Now, to do that, there's a number of techniques. So I'm going to start with one which is actually engineering cells. And this is something that would be very applicable, say, to technologies like stem cells. So we introduce stem cells into the body, we induce them to differentiate in a particular cell type, maybe to repair the heart and add cardiomyocytes, or to add insulin-producing cells in the pancreas. But we actually want to track what's going on. At the moment, there's pretty much a black box. You do a stem cell therapy experiment, the cells go into the body, and you wait to see if there's a biological result. You have no idea what's happening to them. With this technology, we have the potential to track them every step of their way in the body. So this is an example where you basically just use a promoter, a gene promoter. It doesn't really matter what it is, but you could target it to any particular gene that was strongly expressed. and you hook that to, in this case, an enzyme that is going to activate your imaging contrast agent. And you can put various forms of imaging tool in the same construct. So in this one, we've got an enzyme, thymidine kinase, that we're going to use to activate our imaging agent. And we've got a light emitter, green fluorescent protein, that will give us a fluorescent signal so we can pick it up by a light signal as well. And it's really a very, very simple approach. You transfect the cell with this construct, it expresses the thymidine kinase, and then the imaging agent you use is a small molecule, in this case called FIAU, and FIAU will freely enter cells, go through the cells, and go back out again until it becomes phosphorylated. The thymidine kinase gene that you've introduced into the cell will phosphorylate that reagent, that molecule, and in a phosphorylated form it can no longer get out of the cell. You tag that molecule with a radioactive tag, like a radioactive iodine tag. So now when it can't get out of the cell, your signal builds up, you can image it with a scanner, and you can get a non-invasive imaging signal that is directly proportional to the level of expression of that gene. So you could target this to expression of any gene you like. Insulin gene, a gene involved in heart function, cell proliferation and tumor, cell death if you're trying to kill a tumor, a wide variety of approaches you could use. Just to show you some examples of how this works in an animal model, on the left-hand side, so just rotating, was this agent FIAU. So what you can see there in the orange, little orange spots are some cells we introduced, we transplanted into this animal. And what you're looking at there is not the physical location of the cell, but the expression of a gene within those cells. And on the right-hand side, we've used a different tracer that acts much more as a representation of where they are physically. It's just a marker, a physical marker of where they are anatomically, so we can see location and targeted gene expression in exactly the same picture. And this, of course, would allow you to enormously subcategorize, for instance, in cancer, the type of tumor that was present. Now, for clinical use, we don't want to go engineering cells and putting them into people particularly, so we need another approach. And the approach is to use a targeting component, a small molecule that will specifically target a particular type of cell. And in a disease situation, you wanted to pick out disease cells from healthy cells. Now, fortunately, many disease cells display a totally different array of proteins on their cell surface than healthy cells. And if you can select these and come up with protein with markers that will stick to those specific cells, you've now got a means of tracking them for imaging. So these targeting components can be small molecules coming from the huge libraries of chemicals that chemists have produced over the years. They can be small peptides, growth factors that will interact with specific receptors, for instance. Or they could be much bigger things like monoclonal antibodies or antisense nucleic acids. Basically, the smaller they are, the easier they are to get into the tissues, and the quicker you will get an image. But you take this targeting component and you hook it to an imaging component. And now you can target your imaging tool to a very specific cell population. Now, when it comes to the imaging component, we have a wide range of techniques we can use. We can look at it by SPECT. SPECT use radioisotopes, quite long-lived, so those half-life can be hours to days. It is an ionizing radiation, so you have to be very careful of dose. Alternatively, we can use a radionuclide technique called PET. Now, PET gives us a wide range of molecules we can use, radio-labeled carbon, nitrogen, a huge library of things we can label. And there the half-life is much shorter. It's in terms of minutes to hours. So in fact, we can get very precise, very quick images and clear the radioactive tracer from the body very, very quickly, so there should be very few safety issues involved in this sort of technique. Now here's an approach where we've said we want to target this particular tumor. And this particular tumor expresses a biomarker, which is a cell surface protein called an integrin. And so through this route, we've been able to come up with a small molecule, a very small tripeptide, which can target the specific cell surface marker. And we've tagged that, in this case, not to a radioactive probe, but to a light-emitting probe. And then you put it in a suitable device, and you can see, here's the tumor. And by the intensity of the signal, displayed here as a color change, you can get an idea of the phenotype of the tumor by the expression of these marker proteins on the cell surface. So it gives us an incredible potential to subcategorize disease non-invasively in a way that before we could only do by biopsy and histology. The second example I want to give you is the emerging sophistication of therapeutics. So this is the traditional approach to therapeutics. Your doctor prescribes you a pill. Clinical practice guidelines say that you should get a certain amount of drug, so many milligrams per day. If you give that to an individual, then that dose could either be effective, or it could be sub-effective, it could lack efficacy, or it could actually turn out to be toxic, and you actually suffer from being overdosed for the drug. And yet that drug, according to your kilogram body weight, was exactly what the physician should have given by clinical practice guidelines. The reason we've got this diversity is, of course, that we all actually respond slightly differently to drug metabolism. We metabolize drugs in different ways at different speeds. And understanding that of the individual will greatly enhance your ability to dose an individual appropriately for a particular condition. So this is now looking at the interface of disease, environment, and genetics, because a major predictor of the way that you will respond to what is an optimal dosage of a drug for you will be your individual genetics. And by capturing that information and feeding it into the algorithm, we can personalize the dose of a drug that would be required for optimal treatment. Now, it turns out that many, many drugs are metabolized by similar classes of enzyme, and one of them are the cytochrome P450s. And this particular class of drug, the CYP2s, are responsible for the vast majority of drug metabolism in our body. Adverse drug reactions are the fourth leading cause of death amongst hospitalized patients. So this is not trivial. Getting dosage right it may have a huge impact on hospital safety. 1 in 11 patients will not benefit at all from drugs like codeine because they have a genetic polymorphism, which means that they simply can't metabolize those drugs. They have a polymorphism in the CYP2D6 gene. And so knowing who is actually going to respond to a drug or not respond to a drug is fundamental to choosing this treatment strategy. That same enzyme also metabolizes tamoxifen, which of course is a drug that's widely used in breast cancer. So this, again, is not something you want to mess around with. If you simply got a headache, you could say, okay, we can afford to get the drug right, the dose right. If you've got breast cancer, you don't want to mess around playing around with dosages. You want to get it right the first time. Another example is anticoagulants. A frequently used anticoagulant is warfarin, the blood thinner. The little graph there shows you the variation in what would be an optimal dose for 3 different individuals who have different polymorphisms in the gene for the enzymes that metabolize warfarin. So you could have a 5- or 6-fold difference in what is an optimal dose for you depending on your individual genetic profile. And knowing that profile, which can be done very easily, and you only need to do once for your life because your genes don't change, would give clinical guidance as to exactly what was the optimal therapy dose. And the FDA is now starting to cotton on to this and are starting to alter their labels on drugs to display the risk that they recognize that a particular dose may not be appropriate for a patient based on their individual genetics and the polymorphisms that you can measure and display, and starting to put qualifiers that physicians should really take that into account when prescribing drugs to a particular dose. The third example I want to show you is really the treatment of chronic disease, the management, prevention, and treatment of chronic disease, which is, the healthcare disaster that is on our doorstep at the moment. And this slide really shows the urgency of the situation. It's a projection of the burden of disease of diabetes in Canada looking forward to 2016. And the baseline data was collected in 2000. So you can see that diabetes, particularly type 2 diabetes, which is the majority of diabetes, is still a disease predominantly of the elderly. So here's age groups along here. Here's the person suffering from diabetes or projected to do so in the green. And you can see that it's really a disease of the 50s, 60s, and 70s. But looking forward to 2016, it will become a disease of middle age. And we will be dealing with twice as many people in the elderly brackets who are afflicted with diabetes. And how do we respond to that enormous healthcare burden that will drop upon us? To really put this in perspective, look at the decrease here at around 75. This is people who are going to die. This is the shortening of life that is due to diabetes and our inability to appropriately prevent and treat it. So diabetes on average cuts life short by 13 years, even in spite of all of the sophisticated drugs we have to control it and our understanding of how to prevent it. To make matters worse, in southwest Ontario, our demographics are changing in an alarming way. And this is data that's been put together by our local health district in London, the Southwest LHIN. And what it shows is a projection of distribution of population by age from 2007 through to 2017. You'll notice that, in fact, we start to lose teenagers. We are going to lose relatively young people as a proportion of our population. But we will gain enormously people in the 55 to 64 and 65 to 74 brackets. They will carry with them this enormous increased risk of diabetes, amplifying to the burden that the healthcare system is going to have to deal with. Now, the problem with diabetes, of course, is not dealing with diabetes, regulating blood sugar. Many of those drugs that are used for that are relatively cheap, and of course lifestyle is as effective as almost anything else in type 2 diabetes. The problem is preventing the onset of complications in people with diabetes. So the retinopathy, the blindness, the kidney disease, the cardiovascular disease, the heart attacks, the stroke. And if you look at this economic data from a Finnish group, you can see the mean cost of care of diabetes for an individual in which you have prevented the onset of complications. They have diabetes, but they don't have complications, versus those that have complications and need typically expensive hospital care and drugs. And the differential is absolutely enormous. So look for type 2 diabetes. That is the differential between good control, no complications, and what happens when a patient has complications here in euros. If we look at the complications, they are by far cardiovascular disease. 80% of the complications associated with diabetes you will find in people are in the category of cardiovascular disease, and we know the impact that has both on quality of life and burden on the healthcare system. So it's not only preventing diabetes, it's keeping diabetes well controlled and preventing complications. Now, when do vascular complications start? I'm going to show you some of our own work from London, where we actually looked in kids, and we looked in obese kids, knowing that obesity is a risk factor for diabetes. These kids do not have diabetes yet, but they do have impaired insulin action, so they have insulin resistance. They're on the slippery slope to getting diabetes, and they're that way because they are obese. So we have a number of ways now that we can gain information, again, essentially non-invasively, about the health of the cardiovascular system. And one of them is this technique called peripheral arterial tonometry. It basically is putting a cuff around the arm of the subject, inflating it to restrict blood flow for a few minutes, then letting it go, and then the vessels dilate as the blood flows back in. The speed of dilation of the blood vessels is a direct measurement of the health of the blood vessels and the endothelial cells that line the blood vessels. And it's those cells that line the blood vessels that are often the root of cardiovascular disease. That's where things go wrong first. So the lower the score, the more disease the tissue. So you can see in obese children, even though they don't yet have diabetes, they are already showing signs or early signs of impaired vascular function and the start of cardiovascular disease. And these are kids aged 12 to 16. There's a number of other ways that we can also track the cardiovascular health. No matter which technique we use, we always find when we plot it out, this sort of relationship, it's continuous. So in this case, we're looking at this occlusion technique. So as vascular health deteriorates, there's a straight correlation with increase in body mass index or obesity. So there is no cutoff at which you're healthy and then you're diseased. It's a continuum. Now what that says is that any weight reduction, anything we can do to reduce weight in these overweight kids is going to have a beneficial effect on cardiovascular health. Similar situation with this. This again is a very powerful technique and is non-invasive. It's a 3D ultrasound. So we all know about ultrasound in looking at the unborn baby in obstetrics. You can also look at blood vessel health with ultrasound, and this is a technique that was developed at the Robarts Research Institute in London of 3D ultrasound. So this is actually a carotid artery in the neck of a subject, and you can see in exquisite detail, down to less than a millimeter in definition, the vessel wall. You can start to look at thickening of the vessel wall as a disease process, and if an atherosclerotic plaque starts to appear, you can pick that up and monitor its progression to eventually will block the artery and potentially give rise to stroke. So we can non-invasively and longitudinally track with exquisite detail the health of microvessels and macrovessels. All this information is new within the last few years, and how do you integrate that information in with the traditional clinical parameters of health and the emergence of a disease like diabetes? That's one of the challenges we have where health informatics is going to, I think, produce solutions. Now, the good news about this is you can intervene. So this is a similar group of obese children where we've actually actively intervened in the disease process. And we've done that through lifestyle coaching, very intensive lifestyle coaching with a team, a kinesiologist to devise an exercise regime that they can do and enjoy, a dietitian to advise them on how to live with peers while eating healthy at the same time, a social worker to deal with their issues and worries around this, and a clinician to actually manage their health. So we use lifestyle alone, just a 6-month intervention, or we've been more aggressive and give lifestyle plus a drug, metformin, which is a standard drug in control of glycemia. Now, over that 6-month period, if you look at the body mass index between the start and after 6 months, in the lifestyle alone, the body mass index increased. Now, this wasn't a failure. If they hadn't been exercising, they hadn't had a diet, it would probably have increased much more. But if we actually become more aggressive and include lifestyle and some very safe and well recognized therapeutics like metformin, now the body mass index starts to fall. And this is the delta change over the 6 months. So you actually start to reduce body mass index, real evidence of a reduction in obesity when we take this more aggressive approach. Now, interestingly, a lot is said these days about the value of a waist measurement as an indication of obesity. In this group of children, in fact, there was no health whatsoever. Didn't give you any indication about what was happening to body mass index. So the delta change was very encouraging. If we looked at insulin sensitivity through a technique called HOMA, then insulin sensitivity improved. It went down, both the lifestyle and the lifestyle plus metformin intervention. When we look at actually the biochemistry and the lipids, well, we know that HDL going up is a good thing, and that happened. LDL going down is a good thing, and that happened. And triglycerides going down is a good thing. The lifestyle plus metformin was consistently more effective than the lifestyle alone. So we can get real changes in biochemistry. And out of this type of study, we're able to identify new biomarkers, new lab tests that will give us an indication as to the direction that metabolism is taking. And one of those biomarkers that's very useful in obesity and the risk for diabetes are classes of compounds called adipocytokines. They are hormones actually made by the fat. Many of you, I think, will have heard of one called leptin. It's involved in appetite, so When we eat a lot, we produce leptin and we feel full and we don't eat anymore. Another one is called adiponectin. And what we found is by looking at the ratio of adiponectin to leptin, we have a very sensitive indicator of the direction of metabolism. And so here in this intervention, between 0 and 6 months, we see this ratio change dramatically, more so for the lifestyle and metformin than lifestyle intervention alone, and this shows the delta change. So we've identified a very sensitive biomarker that will tell us which direction metabolism is going in. A new source of information, something else to plug in to the matrix of informatics that will help give the right treatment for the patient. Again, completely continuous distribution between the change in the adipocytokine ratio and the change in BMI. So again, there is no strict cutoff between health and disease. Anything you do is going to be good. Now, it's not as simple as all that because, as you may have picked up, I talked about lifestyle intervention very glibly, as if it was an absolute. And in fact, it's a hugely uncontrolled variable. You think about the lifestyle advice that is normally given. Exercise 30 minutes a day. It doesn't tell you what type of exercise. It doesn't tell you whether that exercise is right for you. And it doesn't tell you exactly what is that going to do to your cardiorespiratory function. In fact, the type of exercise you take and the effectiveness it has very much depends on your personal genetics. And this is a paper, for instance, that shows that polymorphisms in just 2 genes, these PPAR genes, will affect the way the change, your actual response to aerobic physical exercise, your change in fitness, and also your change in insulin sensitivity during a lifestyle intervention for obesity or diabetes. So you cannot simply prescribe a standard exercise regime. That has to be potentially tailored to your individual physiology as well, and you can titrate that by knowing the individual genetics associated with relevant polymorphisms. Similarly here, polymorphisms in one of those adipocytokine genes, adiponectin, will affect how adiponectin responds to exercise. So to interpret a biomarker, you really do need to know what the personal genetics of the individual is. Now, what is very encouraging about that is that in a situation where aerobic exercise does not seem to work particularly well for a lifestyle intervention, something like resistance training does seem to work effectively, and vice versa. So there is every opportunity to tailor an effective exercise regime to the individual, again, based on knowledge about their individual genetics. Now, what I've shown you at the moment is an at-risk group who are at risk of going on to diabetes. But can we further dissect out who is really at risk in that group of going on to diabetes? And that can partly, again, be done by genetics. It used to be thought we could never predict genetically who was going to get type 2 diabetes because it's a polygenic disease involving the interaction of multiple genes. Now with high throughput screening, genetic screening, it's possible to come up with algorithms, patterns of gene polymorphisms, which are highly predictive of who will develop type 2 diabetes. Now in most cases, we have no idea what those particular genes where the polymorphisms are do. And in most cases, there is no obvious linkage to insulin production or insulin action. And it doesn't matter, because as long as we have the pattern and the association with the risk of disease, we can use that as a biomarker and use that then to subdivide populations into those who are particularly at risk and who need the most intensive healthcare interaction. To add one more variable and show you why the complexity of the health informatics system we will have to evolve just in this one example. What I've talked about so far in terms of genetic variation has been true changes in the genetic code, polymorphisms, single nucleotide shifts or substitutions in a particular gene. However, the most variation in our susceptibility to disease is not based on DNA sequence. It's based on something called epigenetics. And I'll show you one example which amplifies that. This is an animal model of diabetes. It's a model where, in a rat, we feed the young rat a low-protein diet. So essentially it's a malnourishment model of the rat while it's a fetus. These animals tend to be born small, and they're sort of like equivalent to growth retarded human infants. And as they grow to adulthood, they develop type 2 diabetes. So if we look at glucose tolerance, for instance, we give a glucose challenge and look at the ability of the animal to handle that, to mobilize its insulin. What we find is as day 85 of life, they can handle glucose perfectly well. By the time they get to adulthood at 130 days, then the animals that had seen low protein diet very early in life are now glucose intolerant. They've essentially developed type 2 diabetes. If you dissect this out, the males have developed diabetes because they get pot bellies, they get visceral obesity, and then they develop insulin resistance. The females become diabetic because they have a failure of insulin production from the pancreas. So actually there's a gender difference in the way that the animal develops diabetes, although diabetes is diabetes at the end of the day. But that sort of variation you see just on a gender basis in these animal models. Now this risk is not genetic, it's epigenetic. And what has happened is that early life environment, that low nutritional environment, has changed the set points at which genes are expressed. And this is done by changing the promoter regions to genes through mechanisms like methylation. If you add methyl groups to them, you can close the gene down and stop it being expressed as highly. And changes to the chromatin structure in the nucleus that actually house the chromosomes. So through chemical alteration, you can close the chromatin down, the transcription factors can't get in to the genes to actually activate them, or you can open the chromatin up and you get much more easily, you get gene expression. Epigenetic programming involves these sort of changes. It's not changes to the genes itself, it's changes to the ability of the genes to actually be expressed at a certain level. Again, we can come up with patterns of epigenetic change, either to chromatin or methylation patterns on the promoter region, so we can quantify that information and we can start to feed it into algorithms. And this actually accounts for most of the individual variation in disease, much more than genetic polymorphisms. To give you a real-life example of how that applies to human health, this is a study that looked at glycemia of the mother during pregnancy, so the ability of the mother to control her blood glucose. Some of those mothers actually were truly diabetic, others were simply tended to be hyperglycemic but not yet diabetic. And it correlated that then, following their offspring, to what was the weight of the offspring at 5 to 7 years of age. And what this showed was a very strong correlation between the risk for childhood obesity and the mother having poor glucose control during pregnancy. So the risk of diabetes is in fact imparted by the mother long before birth. Now a major risk factor, of course, for hyperglycemia in the mother is maternal obesity. And a major determinant of maternal obesity is socioeconomic status. So this is Canadian data from Nova Scotia. Interestingly, it was marketed in the press as socioeconomic status and peripheral outcomes in a setting with universal access to essential healthcare services. It's interesting that some people interpret the Canadian healthcare system in that way. But it made the assumption that access to healthcare was no longer a variable, that everybody had access to appropriate healthcare. And what it then did was to look at the pre-pregnancy weight of the mother and correlate that with income. And not surprisingly, those of lowest socioeconomic status tended to be the most obese pre-pregnancy. Here's another little interesting indicator, I thought, whether you have an RRSP or you don't have an RRSP, and a very good correlation with maternal pre-pregnancy weight. So I won't even begin to go into the factors that would account for this, socioeconomic pressure on the ability to buy quality food, access to quality food, where these people are living in relation to food, availability, etc. There's a whole science just in that. But what this brings out very much is that when we start to look at the variables, we're not just looking at bioinformatics. We're not just looking at molecular and cell biology. To truly get a grip on the variables and the way you would have to analyze them, in predicting the risk of diabetes, intervening and controlling it, you really do have to go all the way into socioeconomics, politics, and every aspect of human health. So we're all familiar with this picture, and this is the sort of thing that Richard Kim, I'm sure, will show you. It shows the relationship or the tools we now have to assess individual genotype through genotyping, through things like DNA microarrays, to couple that with other protein arrays to give you individual protein signatures that are diagnostic of disease, to carry that forward to selective pharmacogenetics, give the right dose of the right drug to the right person, and to subclassify disease so that we can more effectively treat smaller groups of people, ending up with something that often is called individualized healthcare or personalized medicine. But in fact, the real situation is much bigger than that. This is just some of the types of information that we would have to collect if we truly want to have anywhere near an optimal algorithm as a predictive tool for preventing and managing diabetes. Clinical phenotype, parental history, and perhaps beyond, perhaps to the grandparents' health history. Anthropometry, and not just weight and height, but start to get to grips with things like percentage of fat, how much is visceral, etc. Molecular and cellular imaging to start to give us detailed, non-invasive biochemistry. Biomarkers, individual genetic variations and patterns, ethnicity, which we know is a major factor in risk disease. Epigenetic patterns, which are probably more important than genetic polymorphisms. Variables in the physical environment. We know that, for instance, environmental toxins like dioxins are a major feature in increased risk for type 2 diabetes, and socioeconomic factors. So huge amounts of very different types of information that we need to assimilate and mine in order to get the maximum value out of this to help us in combating diabetes. So in fact, the levels of information that we would need to obtain go all the way through from the molecular to public health. And to mine this effectively, we need an incredibly robust and intuitive system that takes advantage of artificial intelligence, neural networking, all of those other tools that you develop in buildings like this to really get to grips with real-life medical health issues. Good news is we can do this. This is one of Famida's models that I'm showing, and it's a model where she has looked at the variables involved in compliance with hypertensive therapy to yield a target blood pressure. So here are the factors, the demographic factors going in: age, gender, ethnicity. Here is the compliance outcomes. Are you taking medications? Are you adhering to diet? Are you adhering to lifestyle intervention, exercise interventions? And these are all of the types of variables which impact this outcome. So this can be modeled. The effectiveness and sensitivity at any one of these variables or combinations of these variables can be modeled to look at their likely influence on the outcome. So the sort of sophistication we need to model these multivariate systems is at hand and can be developed. And one of my messages today is we can do that together. We have huge sources of data input of a quality and sophistication we would never have dreamed about a few years ago. You have some of the technology we need to effectively mine and make sense of this data. So this synthesizes it really, very different types of data: biochemical, kinesiology, nutritional, socioeconomic. We need intelligent analysis that will not replace medical expertise, but will be used as a tool by medical expertise to better support diagnosis, decision, and treatment. So these are some of the mining techniques that we are lining up as potential areas where we think we're going to get value from our data sets. So clearly, creation of predictive models will help enormously in identifying who is most at risk and what is the time scale we need to intervene. Link analysis, looking for connections between pieces of data we would never intuitively think was there. An example being some of the gene profiles that provide risk for chronic disease. You would never guess that those genes were involved unless you saw the patterns. Database segmentation, the ability to subclassify a patient cohort in a way where you can better offer treatment to the individual. And then perhaps most challenging, deviation detection, because what you really want to identify is your outliers, the patients who really are not responding as you would predict. So not only do you have to take the data from individuals into collective databases, ideally you want to be able to break them out again, back from the collective data, back to the individual, and now look at the individual in the sense of they are an outlier, and why are they an outlier, and therefore what is the correct course of treatment for this individual. So how are we going to do this? Well, one thing became very clear a couple of years ago is that there is not a hope in hell that Lawson can do it alone or Western can do it alone. The best approach is one of collaboration. And when we looked around, we actually have all the skills we need within the academic institutions of Southwest Ontario. So we have formed a network, and it's got a brand. It's called the Life Cycle Research Network. It consists of the London hospitals, Lawson, University of Guelph, bringing huge expertise in nutritional science, University of Waterloo, with its expertise in computer science and health informatics, University of Windsor, strengths in cancer research and various aspects of cardiovascular disease, and the Windsor Regional Cancer Centre. Our goal is to create the architecture for multidisciplinary linkages throughout Southwest Ontario, targeting initially chronic disease. So to identify population risk factors, to evaluate the impact of intervention programs, and to translate that learning into clinical practice and policy guidelines. So it's a very functional goal. This is not an academic ivory tower. To do that and to convince others to invest in it, we have to show that we're being innovative, that we're not just trying to do what others have done before and often failed. So what can we do that's truly innovative? Well, we can identify subpopulations of individuals for optimal treatment of disease or disease prevention. So the right treatment for the right patient delivered in the most effective manner. The interventions need not be pharmaceutical. They could be nutraceutical. They can be lifestyle. They can be educational, or they can be combinations. All could be equal effective, and they can be potentially be synergistic. We can link personal and ethnic genetic risk with metabolomics, diagnostic imaging, kinesiology, nutrition, socioeconomic factors, and our environment. It applies literally from before birth, from fetal health through to healthy aging, hence life cycle. And we can create innovative cohorts. The one big limitation of working in a hospital-based research institute is that your patients come into a hospital. They are, right from the start, you are taking a subsection of the population. What we hope to do through this network is actually to extend our ability to recruit cohorts out into family practice centers, long-term care establishments, workforces. We've already persuaded one of our hospitals, London Health Sciences Centre, to consider putting in a fitness centre as an employee benefit, but which would give us the ability to use our hospital employees as study cohorts. We can also then take in pediatric cohorts, rehab cohorts in our own academic fitness centre. We're talking to employers about using their employees as cohorts for health maintenance, for obvious attractions, for reducing sick time, etc. We have our own population studies. For many years now, we've been following the community in Walkerton, who was stricken with E. coli contamination resulting in several deaths. Well, the long-term health of that community following that E. coli exposure is a complete project in its own right, and it's turning out to be extremely interesting in the way that we can longitudinally track the health of that community, those who are infected, but also those who are not infected. It's our own mini Framingham study. And we have a longitudinal birth cohort. So we are tracking every individual who is born in London, looking at maternal health, fetal outcome, and then cross-referencing those to databases of child health. So we have a longitudinal assessment of what are the root causes of chronic disease emerging in childhood life. This is a partnership between academic centres and the regional healthcare providers. It's not been often easy to work with the Ministry of Health for research projects. With this sort of concept, it becomes much more easy. And in fact, we've already had examples where our local LIN, our local health authority, has actually attracted substantial money for health system development projects by using our research ideas. So it really is turning out to be a true partnership between the health provider and health researchers. Diverse sources of data entry have to be taken in, as I've shown, but ideally we need to manage those in shared databases. So we need the ability to create databases where data is emerging from patients all the way through, from Windsor through to Guelph and Hamilton. With that sort of database, we can tackle really large regional healthcare initiatives as research studies if we have the appropriate informatics tools to do it. We can track and monitor incidence and prevalence of chronic disease. We can impact the approaches of intervening proactively in preclinical stages. And we can evaluate ongoing changes in health systems design. So as our health system evaluates new ways of delivering care, this can be the tool that actually evaluates whether they were successful or not. Why chronic disease? Well, because if you target cancer, cardiovascular disease, diabetes, dementia, and respiratory illness, you've actually covered 99% of all chronic disease in Canada. And in terms of health costs, almost 70% of the direct health costs. Why here? Why do it in Southwest Ontario? You know, maybe they could do it better in Toronto or McGill. Well, partly because we have existing strengths. We have internationally recognized groups already working in cancer research and metabolic diseases and neurosciences. But we also have national centers of excellence in imaging, in pharmacogenomics, in nutritional and metabolic studies at Guelph, and in health informatics and database management here in Waterloo. Finally, our region, our population in Southwest Ontario is essentially 2 million strong and is a microcosm of Canada. If you look at the demographics, we are a microcosm of Canada. Many marketing agencies come and test their products in Southwest Ontario because they know it will be representative of the rest of Canada. So we are the right place to actually sample the Canadian population for innovations in health. So these are our initial themes. We're going to concentrate on cancer, cardiovascular and metabolic disease, neurosciences, neurodegenerative diseases, but then with two regional cores. One is going to be the ability to set up clinical trials management such that we could run region-wide clinical trials with patients being recruited all the way from Windsor through to Hamilton. The second will be a system-wide health informatics, which has got to be incredibly sophisticated to make the most of the types and variety of data that we're going to be able to input. The first stage of this is a physical presence for this network. So our first kick of the can is at the Canada Foundation for Innovation, and we have a very large grant funding for, and the Research Hospital Fund for infrastructure. So we're seeking over 100,000 square feet of new clinical research space around the region, from Windsor through to Guelph. And it'll include a number of innovative possibilities. So a clinical investigation unit. This is going to be managed by Richard Kim in London. It's going to enable us to combine genomics, metabolomics, and personalized therapeutics. And it's going to be able to do phase 1 and 2 clinical trials. So we'll be able to be right at the very start of evaluation of new classes of drugs. We're creating a Center for Healthy Living out of Western, out of the Faculty of Health Sciences, that will concentrate very much on pre-disease and preventing the development of chronic disease. Our headquarters for gathering the actual data will be the Network Centre, which is going to be at University Hospital in London. Although the actual mining of that data, we expect the lead will be taken by Waterloo. That this is where the innovation in terms of algorithm design and methodology will come from to make the most of these data sets. We have units specializing in children's health outcomes, in mental health, and really all of the elements we need for those themes. With this one grant, we're seeking to provide adequate physical space to house it, bring people together in right configurations. Geographically, this is what our database is going to want to look like. So we're going to have to put a firewall right around these data sets, from Windsor through to Guelph, and we have individual investigators in Hamilton who are participating as well. So a number of issues. We have to have cooperation between our research ethics boards. We have to have accreditation such that the protocol going into any one will be accepted by the others. We have to be able to secure data and patient confidentiality within this integrated data set. So these are not trivial issues. So finally, at any one of our units, be it London or Windsor or Waterloo or Guelph, we will have what we're calling a Learn Lab. And the Learn Lab will be a clinical research unit with outputs that will enable research translation and will guide health policy formulation. The inputs to that will vary. This shows London's. London's will have strong input from preclinical, from basic sciences, from imaging research, from community medicine, and from health informatics. Guelph's will look different. Guelph will have very little imaging, while we have strong nutraceutical sciences. Waterloo's, again, will look differently. It will be very strong on the health research informatics. But combined, we will actually cover each other's deficiencies, and together we actually look like a very competitive academic unit. So overall, I think what we're trying to do is get a whole new dimension on integrative research, which can only be enabled through health informatics. The data I've shown you has come partly from Lawson scientists like Famida, from Richard Kim. The imaging data comes from Tingley and Frank Prado, and the metabolic data with childhood studies comes from Cheryl Clarkson and Fareed Mahmood. So I hope that I've given you a flavor of the sort of dilemma we're in. So much data, so many issues in how do we make the best use of it, and hope I convinced you of our conviction that the only way we're really going to succeed is pooling our resources within Southwest Ontario. Thank you very much.
I just want to grab the microphone down here. So. Okay, just so you're ready. Now's an opportunity. Thank you, David. Very, very, very interesting. An opportunity for people to ask questions or make comments. I'll start and see what you...
I'll also invite Famida to join in questions.
Yeah, and also Famida. Also, maybe Famida, you may have a few comments to start it off. After hearing what he said, you might want to reverse half of it. You never know. Good.
I guess just to support what David is saying, that we realize for these types of collaborations that we need to be able to work, obviously, where the expertise are. And some of the slides that David showed you are from the EU, some are from the US. And I think what should be quite compelling for us to move forward is to see that other people are already doing some of this type of research in bioinformatics, et cetera. So instead of each of us developing our own facility, it makes sense to actually capitalize on each other's strengths.
And related to that, are there any models, either of you, that we can use as guidance in how to put something like this together? I mean, I've seen things like this before, but where are the models that you respect, with the experience you've had?
Okay, we can probably both give a perspective on that. There are models. I mean, the Mayo Clinic is a superb model of applying health informatics, but that is a single institution where it's relatively easy to do it. There are relatively few examples of multi-institutional approaches to this. There is one in the States. I think South Carolina has taken a similar approach. But really, we're breaking ground in Canada. I can't think of any Canadian equivalent that is trying to do this as a multi-skill, multi-centered network.
Right. So David's right. There's centers in the U.S., and I think the NIH has recently funded a number of translational units to focus primarily on the epigenetics, and then USC's or San Diego. But we've also looked at the landscape in Canada, and there are no other centers within Canada at all that are actually working on such a tight interdisciplinary infrastructure. Most are focused on one particular area.
Right here.
Just to be the devil's advocate, You emphasized, and it has been done quite widely in clinical trials, the use of biomarkers as a proxy for processes that you want to reverse or reduce the progress. We've had, I think, a worrying thing with the Zetia publications very recently. Finally, the trials were completely revealed, and yeah, it reduces the LDL, but it doesn't do anything in terms of what you really want to do, which is to reduce cardiovascular disease. In fact, it seems to make it worse. So how do you, I mean, I'm sure that your project is going to be an attempt to use multiple markers to try and find out which ones are the really reliable markers that are related to the actual physiological processes that are inherent to the disease. That
there's ultimately only one gold standard, which is have you changed the course of the disease? It takes time to get to that, particularly in chronic disease. You may not be able to come to that conclusion for 15 to 20 years. Funding agencies and governments do not have that amount of patience. Therefore, we have to use biomarkers. But we shouldn't ever get into the trap of thinking that the biomarker is actually 100% surrogate for the disease process, because we both know it's not.
Yeah, and who knows? I mean, it may be the LDL in the long run may be incidental. That there's really something else that's tied to the same process that you're manipulating with the statins and a variety of other things that really is what is modifying the progress of the disease.
I think that's a great example. So in your example, we know that the data was actually available in 2006 but was suppressed, right? So what we're proposing is that as data becomes available, you actually have it in a database that you can share and look at on a proactive basis. Because the key point is that, in clinical trials, we're trying to determine what the number needed to treat are. But in real life, there's a number needed to harm. And so to be able to detect the harm early in the game, which means that we can actually intervene. So you can use it both ways.
Anybody else put your hands up and we'll get a microphone to you. I'll ask so many questions I want to ask, I don't know which one to ask. But one issue is the sheer organizational issue. Yeah. I'll think of it from two directions. One, the difficulty in actually getting institutions to work together on this kind of environment and the technology needed to facilitate that. But another issue is the issue of the ability of the health system to really support this and the effectiveness or ineffectiveness of the LINs in that regard. It's a kind of an open question, but can you address any of that?
There's a number of levels of how do we deal with the governance and management. So first of all, at a governance level, I think we're in fairly good shape. This network now has a board. On the board are the VP researchers of each of the constituent universities, the CEO of the London Hospital, etc. All of them committed, including the VP research of Waterloo, to following through on this vision. But of course, it's not VP researchers who actually do studies. It's collaboration between investigators. So equally important is the networking that we can achieve within the region to get the right researchers talking to each other. And there's no easy solution to that. As you know, it's sometimes difficult to get people in the same university talking to each other when one's at one end and one's at the other end. So you have to work continuously at this interaction. But from what we've seen already and the partnerships that are already formed and the joint grant applications that are going in, there is the critical mass to enable this to happen within our region. And yes, it will not all be there at the start. We will see examples of where partnerships flourish, and other ones will be attempted and they will go nowhere. But it will be a continuous momentum. Of course, at the end of the day, there's nothing that enables that encourages investigators to work together other than an opportunity, either dollars, which usually works pretty well, or raw material. And the raw material we will have here is access to data sets. So as Famita said, the idea is that these data sets will be lodged in a repository which will be accessible to all serious qualified investigators. These will not be resources that are clutched to individual or institutional chests. The idea is they will be shared as resources where a variety of questions can be formulated out of the same data set.
We've seen it necessary in these kinds of environments, though, also to support the intercommunication, the real interaction among the researchers. To what extent is that in your vision of this? In other words, a technological infrastructure that supports, we usually call it communities of practice, that kind of thing.
Yep. I mean, it's maybe for me it's better to answer to this one because she's actually started this process.
You know why you brought her?
Yeah.
Well, he actually needed a ride. No, I'm joking.
So it's your monster.
Yes. Anyway, so we're looking at this from multiple levels. The first step is to actually set up, for example, a website where we can all post information, including documentation. So we've done that. The second is using a mechanism for communication. So something even very simple like SharePoint, where we can all go in, work on documents, set up meetings, etc. The third is going to be setting up a virtual organization. So can we have labs that are maybe smaller than this one, but where we can all communicate with one another. We can look at each other's screens. The images that David was showing, we can potentially look at together and do some brainstorming. We can apply each other's algorithms, etc. So that's step 3, and that's the direction that we'd like to move in, is to actually be virtually linked to our key partners, particularly in Learn, to facilitate some of the research questions. And the research questions, just to be clear, happen at multiple levels. So it's not just the clinical researchers or the basic scientists. There's computer science research, there's mathematical research, etc. So there are levels of research and development at every level of this interdisciplinary team. Does that help?
Your other question was about integration with the healthcare system, with the provider. And it was very, very difficult when we had a centralized Ministry of Health. What has happened with the evolution of LINs is that LINs are now jockeying to be the most innovative LIN, to adopt, to go into beta projects, and there certainly is money out there. And the example I gave was, the Southwest LIN, which covers everything from Port Stanley up to Tobermory, with London in the middle. Was recently successful in getting, I think it was $8 million out of the provincial government to pilot a new model of delivering healthcare for diabetes. They used as the rationale for that the emergence of this network and the ability to ask appropriate questions, to analyze the data, and to apply the data analysis that we were promising through this network.
Excellent. Very interesting. Other questions? Over here.
Can you amplify a little bit on primary health care and the role of primary health care that you see in this study?
It's considerable, because only then will we truly get to the cross-section of patients to gather the data we want. We have some pilots in London that are doing that through research networks of primary healthcare providers and family centres. Dr. Stuart Harris, for instance, has a network of I think almost up to 50 now family practice centres, all of which who are involved in research projects, all of which who can input data to a central database, asking in this case questions around diabetes diagnosis and ongoing management. So it's going to be a gradual hub and spoke network. We are setting up the hubs, but the idea is that networks will develop around those. That'll include primary care. It'll also include any other environment where people are receiving health care. Long-term care establishments is another one. One idea that Famida and I are actively pushing is the partnership of pharmacists potentially in this. Most diabetics or people with diabetes have infrequent access to a diabetes educator. Only spasmodically will see a diabetologist in hospital, but all of them go to the pharmacy. So we're better to start to collect data and enter it into these sort of databases than to engage pharmaceutical chains in the process as well.
Another devil's advocate question. You have been talking about the compiling of large databases of what will have a lot of personal information in it. Are you dealing with the Privacy Commissioner and those folks? Because I've
do that proactively. We know the Privacy Commissioner very well in London. We have had many situations where we've sought the advice. One advantage is that London and the Thames Valley region now have almost 10 years experience of pooling data between hospitals. We are towards the front of the province in rolling out electronic patient records. We have experience in linking records between different hospitals within a geographical area, and maintaining privacy according to the legislation. So I'm not saying we figured out all the answers, but we do have experience that others haven't in how to approach it.
It's not easy.
I know.
Unfortunately, I guaranteed David, because they have to get back to a meeting, that we would end right now and give enough time for him to get out of here by 4:30. So I want to cut this off. I also want to point out that I saw Eric in the background there, one of our stem cell researchers. I'm sure we'll want to say hi to you. Yeah, you know each other real well. And but I want to thank you, David, for a very, very interesting talk. That I wish we had an hour now to go through lots of questions. But thank you. Very informative, very interesting. Look forward to working with you.
Thank you.
Here's a small gift. Thank you so much for coming today, and I'm sure that we'll have many active discussions following this. Thank you all for coming today. For those of you here and online, please come back and see us again on February 27th when we'll be interacting with Dr. David Kim. Have I got that right?
Yep.
Richard Kim. Richard Kim from
You can call him all David.
Or Peter or what. Anyway, Dr. Richard Kim from the University of Western Ontario. Thanks so much. All right. There you go.